Right. Okay. Mapping with somatic cell hybrid panels. Now, again, I mentioned, I think one of the first few videos is that one of the challenges in analyzing the human genome is really to look at or to obtain individual chromosomes. In humans, in each cell, you will have 23 pairs of chromosome. So one of the first challenges is to separate them so that you can actually work on a single chromosome without as I mentioned, if you have 23 different jigsaw puzzles all lumped together, one of the first things you want to do to solve it is to make sure that you have the pieces of the right puzzle, right? So you need to separate, okay, this is puzzle number one, puzzle number two, puzzle number two. So this is what they're doing with the human chromosomes. So they need to separate it. So one of the methods that they use is this, given in this picture. It's called the establishment of somatic cell hybrids. Now, essentially, what it does is that you fuse two types of cells. One is the human cell, the other one is the rodent cell. So what you have is a heterokaryon. Karyon means karyotype, karyon means nucleus. Heterokaryon, you mean a mixture of chromosomes in a fused nucleus. But then something funny happens. What as the hybrid cell makes copies of itself, what tends to happen is that the human chromosome gets chucked out, right, randomly, and this process continues as the cell divides and you will until it becomes stable. And at that point of stability, you'll see that each cell will probably contain about 1, 2, or 3 human chromosomes only. So you keep at it, and then you keep enough clones, and then you'll find that you will have a collection of uh, hybrid cells, somatic cell hybrids that contain different human chromosomes individually. All right, so this is somatic cell hybrids. Later progresses in and advances in this is that instead of having single chromosome, they use chromosomal chunks, even smaller pieces that they stably reproduce using this hybrid cells. All right, RH mapping is radiation hybrid mapping. Um, what this does is that once you have all these cells or colonies, and then you sort of put them in a 96 well plate. So on a 96 well plate, that means you have 96 clones, and then you start to see which one overlaps with overlaps with which, and then it is actually, when you deal with so many different colonies, there's actually a robotic system that does this. You have all these plates, and all these clones are all ready to be analyzed, and they're kept, and then they use a robotic system to actually retrieve the clones that you are interested in. All right. Um, another way of doing it, that previously what you do is you just marry different chromosomes. Now the other way of doing it is, again, you start off with somatic cell hybrids, and if you've done this on your cytogenetics, what you do is that you drop it on the oh no, what's the name, slide, and then you let it dry. If it dries up, right, you will start to see shrinking and shrinking and shrinking of the cytoplasm, and it may shrink enough that it only covers one chromosome. That can be taken out and cultured, and this is what you get: microcell. Right? Maybe my explanation is not clear enough. Have a look at at the net, have a look at that, and then you probably be able to find a picture or video of what a microcell is. But essentially, these are clones that have specific individual chromosomes, right? Don't lose track. Why we're talking about this is that the human genome contains 23 pairs of chromosomes. You want to make clone contigs, so one of the first steps is to separate the chromosomes, all right, into different resources. So I know if I want to work on chromosome 7, I can just call some lab that does all this separation and say, excuse me, can you give me clones that contain chromosome 5. If I want to work on chromosome 7, I can just call some lab that does all this separation and say, excuse me, can you give me clones that contain chromosome 7? And then when I have that, then I can work with chromosome 7, build context and so on, right? So this is the process of how you analyze human genome. And the next section, see, I mentioned STS content mapping. We looked at the chromosome, we looked at the overlapping fragments, and these overlapping fragments are based on, if you know the STSs, the markers, so you can do STS content mapping. And what do you clone this chromosome in? I'll get back to you on that one on the next video.